As usual, there's a series of brushing up topics. This week's are pretty important. They ask you to go back and revisit the structure of a protein and the ideas of monomers and polymers. Those are going to be critical to your understanding in this chapter. So our first section here is going to be to look at the structure of DNA and RNA. Note there is identification in this, which means that you will have pictures in your Blackboard exam and be asked to identify all of the parts listed on this slide. Let's get started by talking about what the jobs of DNA and RNA are. Often when you ask somebody what's the function of DNA, they say really big picture philosophical things like it's what explains life and it's what makes us who we are. But what it is much, much, much more specific. And I want you to use the specific definitions and think of these as these specific definitions for the rest of your life. Pretty straightforward. DNA contains genes, and those genes are instructions to make a protein. Go back to chapter 3, revisit what a protein is if you need to. So DNA has genes in it, and the genes are instructions to make a protein. RNA then takes those instructions and actually makes the protein. So that's the two big ideas here. We're going to look at how to make a protein using DNA and RNA, and we're also going to look at how we make a copy of DNA when a cell divides. So let's start working through the identification of DNA and then of RNA. I've split up the identification into several slides, so we'll build up the ability of you to identify all of those items of DNA and RNA. So the first thing to remember is that DNA is what we call a polymer, and the polymer has a specific name, a polynucleotide, and DNA also has a monomer because all polymers have monomers, and in this case the monomer is called a nucleotide. If you don't remember what a polymer and monomer are, please go back to chapter 3 and look those up. So the nucleotide, excuse me, the nucleotide of DNA, which is the monomer of DNA is called a nucleotide, and it has 3 parts to it, called a nitrogenous base, deoxyribose sugar, and a phosphate group, and we're going to look at all 3 of those right now. So this slide shows the nitrogenous bases that are in the nucleotide or the monomer of DNA. So I don't want you to learn any structural formulas. So even though I'm showing them in the slides, you're not going to ever have to sketch or identify these structural formulas. Just want you to know the names and the single letter abbreviations of the 4 nitrogenous bases in DNA. They're listed there: thymine, cytosine, adenine, and guanine, and they all actually are referred to universally by their single letter abbreviation T, C, A, and G. Something else that's critical to know is whether the nitrogenous base is referred to as a pyridine or as a purine. So the pyridines are here on the left, and that's T and C, and the purines are here on the right. They are A and G. Notice that the purines both have 2, apologize while I learn how to sketch, have 2 rings in them, and the pyrimidines, the longer word, only has 1 ring structure in it. So pyrimidines is the long word, has 1 ring in it. Purines are the shorter word, but has 2 ring structures in it. Feel free to laugh at my ability to sketch. So nitrogenous bases were the first part of the nucleotide or the monomer of DNA. The second part is a sugar. It's called a deoxyribose sugar. Long fancy name. It means it doesn't have oxygen in one very specific location in it. I'm not worried about whether you know that. Again, you don't need to learn this structural formula. I just want you to know the name of the sugar is deoxyribose. And when you're looking at these structures in identification pictures, it's going to be hard to know what the sugar is. So I think this O in the ring right here helps you to understand that this is the sugar, and it's not one of the nitrogenous bases, which had those single and double rings that all had nitrogen in them. The last part of DNA, the last part of the nucleotide, the last part of the monomer is the phosphate group. Phosphates are functional groups. You learned about them in chapter 3. So you should be able to recognize this because of the phosphorus in it. And go back to chapter 3 if you don't remember what a phosphate group is. So in this slide, we're going to put all of the parts of a nucleotide together to make the nucleotide. As you can see on the bottom here, this whole structure put together is a nucleotide. So here we start with our nitrogenous base. It could be any one of the four. It's only one of the four at a time. And again, I think I may not have pointed out, you know it's a nitrogenous base because there's nitrogen in it. And in this case, it's thymine or T. The next connected in a covalent bond to it is the sugar. And again, you know it's the sugar because of that O in the ring. And then lastly, you have the phosphate group covalently bonded to the sugar, and you know it's phosphate group because of the phosphorus. So together you have a nitrogenous base, a sugar, and a phosphate, and that all three together make the nucleotide, which is the monomer. It's still just the monomer of DNA. So now in this slide, we're going to put all those nucleotides together to make the polymer of DNA, which is DNA. Let's start with our picture on the right, and I'm going to circle, let's hope I can do it here, let's circle one nucleotide. That is one nucleotide on the bottom right, and first of all, you can see this has the G in it. This is the nitrogenous base. The blue is the deoxyribose sugar, and the yellow is the phosphate. So another way they designate all these parts of the nucleotide is to put the letter of the nitrogenous base, make the sugar blue, and make the phosphate yellow. That's very commonly done. So then what we do is we take the nucleotides and we just stack them. We just stack them up from top to bottom or bottom to top, and we have a nucleotide, and then another nucleotide, and then another nucleotide, and you just keep stacking them up and you get a polymer of DNA, sometimes called a polynucleotide. Now that's a pretty complicated way to draw them, and it's too much work, so in science we like to simplify when we can. So this figure on the left is a simplified version of that. We use flags with the single letter on it to represent the nitrogenous base, and then the sugars in blue and the phosphate groups in yellow. And again, you can see the nucleotides are stacked up from bottom to top or top to bottom, and that makes a polynucleotide called DNA. There is the structure of DNA. So the structure of RNA is very similar. You're going to see that it's almost identical except for a couple of differences. So the monomer is still called a nucleotide and the polymer is still called a polynucleotide, but now we're talking about RNA. In RNA, we're going to see that the sugar is different, and we're going to see one of the nitrogenous bases is different. So let's go through that. Here is the 4 nitrogenous bases in RNA, and the circle on the bottom right shows you that one of the nitrogenous bases has been changed, and it's no longer thymine or the T. It is now uracil, or which is used as a U, a capital U. So uracil is a capital U. I'm using my right hand and I'm left-handed, so that's why my writing is pretty bad. So same three nitrogenous bases, A, G, and C, and one new one, uracil or U, and do not include thymine or T. Now we have a sugar. Again, we know it's a sugar because this zero, this oxygen up here in the ring structure. And this one's called a ribose sugar. It has an oxygen in it down here in that pink box. It's not there in the deoxyribose. I don't care that you know that. Just want you to know the name of the sugar is ribose. And it also has the same phosphate group. So you put them together in exactly the same way to make the nucleotide of RNA. So you have your nitrogenous base, you have your ribose sugar, and you have your phosphate group all covalently bonded together to make a nucleotide in RNA. And then you stack the nucleotides together and make the polymer, the polynucleotide. And again, you can see they're using in this figure that same idea that the flags sticking out are the nitrogenous bases. And then we have the large part of this stack here is alternating with a sugar in the dark blue, and the light blue is called is the phosphate group. I think I neglected to tell you that this long thin strand that goes up and down in the DNA that does not include the nitrogenous bases is called the sugar phosphate backbone. The sugar is always the longer piece and the phosphate's always the shorter piece. So there we have RNA. Now DNA has one more, if I might say, twist. And if you know the structure of DNA, you get that joke. DNA has one more twist to it that RNA does not have. And there's an identification issue here. So using a figure in the book, we're going to look at the fact that DNA is really two polynucleotides, two polymers hooked together in the middle. So in this figure in the center, let's start in the center, you can see on the left we have one polynucleotide, and on the right we have another polynucleotide. Now each one of those is called a strand, S-T-R-A-N-D. Each one is a strand. So we have a strand over here, and we have a strand over there on the right. Now the strands are held together, and you see dotted lines here in the middle, and they're labeled a hydrogen bond. So it's a type of chemical bond. We did hydrogen bonds. I think that was chapter 2. Chapter 2 when we talked about the properties of water and that water forms hydrogen bonds. Well, DNA can form hydrogen bonds between the two nitrogenous bases that are on the left and on the right. So the hydrogen bonds hold the left and right strands together. When the two nitrogenous bases are held together by a hydrogen bond, and I'm putting a box around that right now, that is called a base pair. It's two nitrogenous bases held together by a hydrogen bond. Now notice there's always a single ringed structure, nitrogenous base, a pyrimidine, that's hydrogen bonded to a two-ringed nitrogenous base, the purine. So in the hydrogen bonds, it's always a purine with the pyrimidine, and furthermore, it's always C to G and A to T. That is the rule of life, that in DNA, the nitrogenous bases that are held together by hydrogen bonds that hold the two strands together, it's always C to G and T to A. C and G can be on left or right. T and A can be on the left or right. Now we put the two polynucleotides together, but we take it one step farther. You can see, let's look on the left here. You can see the two strands with their hydrogen bonds being held together. It's also twisted into what's called a helix. It just means it's spiral. So it's twisted into a helix shape. And therefore it's called a double helix. Double because there's a left and a right strand, and helix because it spirals. So that finishes up the discussion of the structure of DNA. There are 4 practice slides in this PowerPoint for you. They're on the next set of slides. I would strongly suggest you do those practice questions. You'll see similar ones on the test. And if you want me to look them over, you can email them to me. I'd be glad to give you some feedback. I'm not requiring it though, so it's not being collected or graded. Okay, so now we have our last topic in this narrated PowerPoint, and it's a critical one. And I want to be clear what we're talking about because this chapter has 2 big topics in it. And this is the first big topic. Now that we've done the structure of DNA and the structure of RNA, we have 2 big topics left. And the first one is how do we make a duplicate or a replicate of a full DNA molecule? This is what happens during mitosis and meiosis. We learned in mitosis and meiosis that when we get new cells, we make a full copy of the full homologous chromosome. So, and then those each go to their own cell. So we have to be able to make a copy, an exact copy of the DNA in mitosis, and except for crossing over, we get an exact copy in meiosis. So this section, this first big section we're going to do in this first narrated part of the PowerPoint is going to talk about how we make a copy of an entire DNA molecule. So it's called semi-conservative model of DNA replication, and we're going to walk through the steps on how that happens, and then there's a learning objective that will help you understand whether or not you are getting how this works. So let's get started on it. It's something that you may have to come back and listen to a couple times and read about and think about. I'll try to walk you through it as carefully as I can. So in this picture, you're looking at the double helix of DNA. I'm going to come up here to the top, trying to put my mouse over it. And you can see that's the polynucleotide on the left, polynucleotide on the right. You've got the hydrogen bonds in the middle holding the nitrogenous bases together. That's DNA. Now, when we copy DNA for mitosis and meiosis, we need to make both the left and right sides have to be copied. You have to have a full duplicate. You don't just copy the left side so that you're left with one strand. You have to have 2 strands in the new DNA. So the first thing that has to happen, I think, is pretty obvious and is not shown in this picture, and I couldn't really even find a good picture of it or an animation of it. But the DNA is coiled. It's in a spiral form, and it has to untwist. So that's not shown in this picture, and again, I don't think it's shown very often in any of the pictures. But the DNA first is going to just untwist and become more like a ladder so that we can break it open and start to copy it. So it's going to untwist. Now, the second thing that happens is then it's going to open up, which means the left and the right side, I'm trying to show with my mouse here, are going to split open. So just in this one small region, the left and the right side split open. Then each side is copied, and you get 2 new copies at the bottom. We're going to do a little more detail on this now. So the hydrogen bonds are broken. The left and right strands split apart. The left strand gets a new side, right side to it. The right side gets a new left side to it. And then they completely split apart, and you have a full new copy of DNA. Okay. So in this graph, we're going to show you, I think this slide shows it a little better, a little bit easier to see than the previous figure that was in the book. We're going to start over here on the left. This is a single strand, single let me start over. It's a single molecule of DNA. You have the left strand and the right strand. The nitrogenous bases are being held together by hydrogen bonds that you can't see. But this is just a normal molecule of DNA. And it's already been untwisted. So it's been untwisted, and we don't have to worry about that step here. Then it unzips. It's the left and right strands separate. It unzips. So you can see now the left and right side, the hydrogen bonds have been broken, and it's been unzipped. Now what happens is the left side gets a new right side, and the right side gets a new left side. So the way you get a new right side is that nucleotides are added, and they're always added so that A goes with T and C goes with G. So at the top one here, you see the A, it gets a T, C gets a G, G gets a C, A will get a T, and T will get an A. And we get the nucleotides added, they all bond, and then this entire structure will be a new strand, a new piece of DNA. The same thing happens to the right strand. And then you can see on the right side that we have now the original. We have the original left strand here, the original right strand here, and the copies added to it. So now we have a new replicated DNA. So DNA we've talked about is pretty long. And pardon me, I just went back past a practice problem, so make sure you do that practice problem. It will show whether you understand how the left and right side split, and then the left side gets a new right side, and the right side gets a new left side. So do that practice problem. Okay, so DNA is pretty long. And in each piece of chromatin, it can be tens of thousands of nitrogenous bases in order. And so you have to replicate the whole thing. And it doesn't just start on the top and go all the way to the bottom, or start on the bottom and go all the way to the top. What it does is it actually copies the strands at multiple places all along the strand of DNA at the same time. So looking at this figure, let's look at the top, the parental DNA molecule. There's our DNA, and you can just pretend the bottom blue line is the left strand and the top blue line is the right strand. That whole thing has to be copied. But what happens is at multiple places along the DNA, shown in green here, the copying starts at those places at the same time. Now the place where it starts the copies all along the DNA is called the origin of replication. So that's the point where we start a copy, and then this next one, another origin of replication. Now another origin of replication. So they all start copying at the same time. So it untwists, it unzips, and you see the origin of replication, and it makes the copy going in both directions away from that origin. So you can see it's gonna, these black dots are the origins, and it makes the copy in both directions. And it does it at every origin of replication until the entire length of DNA has been copied. You can see that it's starting to meet up here, and at the bottom, finally, you have a full new copy of a DNA molecule. Now, I want you to not mix this up with homologous chromosomes. Homologous chromosomes are one chromosome from mom, one chromosome from dad. Each of them is really just a long length of DNA, and each of those has a left and a right. So each homologous chromosome has to go through this replication process. So this is just, say, mom's chromosome, and then it has to happen with dad's too. So don't mix up the left and the right side of a DNA DNA molecule with the homologous chromosomes from mom and dad. Mom's chromosome has a left and right DNA, and dad's chromosome has a left and right DNA, and they each have to go through this replication process. So something that happens in these bubbles, these where the origin of replication starts the copying, and then the place inside of there where it's copying is called a bubble. It turns out that the direction the copying occurs is dependent on whether you're working with the left or the right strand in the DNA molecule. So this figure now labels the strands at the top and the bottom. Let's look at the left one. You can see that the top here is called the 5' end, and the bottom is the 3' end. The 5' end is the end that has phosphate group on it, and the 3' end is the end that has the sugar group on it in the sugar phosphate backbone. So if you start with the sugar, wherever you see the sugar, it's the 3' end. Wherever you see the phosphate, it's the 5' end. And you can see in this one on the left, the 5' end is on the top and the 3' end is on the bottom. Well, the right strand is always exactly the opposite. So in this case, then the 3' end with the sugar in it is going to be across from the 5' end of the left strand. If we go down to the bottom, we'll see the 5' end with the phosphate group is across from the 3' end with the sugar group. So one strand will go top to bottom 5' to 3', and the right strand will go top to bottom 3' to 5'. They're always opposite to each other. So whenever you get a new picture, make sure you look at the picture carefully so you know what you're looking at. Here in these blue lines is a molecule of DNA. You can see there's two lines. One line is the left strand, and one line is the right strand. It is not, again, homologous chromosomes. That's a completely different idea. We just have one molecule of DNA inside one chromosome, and it has, you can see that the 3' end and 5' end of the top strand, which is typically the right one, and then the one on the bottom goes 5' to 3'. And what happens is we had that origin of replication that is not shown here. I'm just going to squiggle it in here. The origin of replication is where that horrible red mark I'm making is. And what we have is the 3' end of that strand of DNA. So for the 3' end, the copy of the DNA can go straight down it. So it goes straight down it from 3' to 5'. And it just keeps adding bases and phosphates and sugars, and it just makes that new strand of DNA. And it goes from 3' to 5' on the original. No problems. And there's an enzyme that does that, and that enzyme is called a polymerase. And the word polymerase is over here on the left. So the polymerase makes the new strand, and it goes from 3' to 5'. Now if we go to the other strand, because it's organized in the opposite direction, to go 3' to 5', it can't do it because the polymerase can't fit into that small space in here. So what it does, it actually starts further down, goes 3', and it always has to go 3' to 5'. So it starts here, and it starts actually farther along the strand, makes a small snippet of the DNA, and then it backs up, and then it makes another small snippet, then it backs up, and it makes another small snippet. So it makes small pieces at a time, and until this gets opened up and it can fit in there. So then you've got all these three pieces that are not connected. And this enzyme called the DNA ligase then puts those pieces together. So you can see on the bottom the overall direction of replication. is always from 3' to 5', even though the strand that runs the other direction, it does it in snippets and backs up and then connects the snippets with the ligase. So the strand is made with the polymerase, goes from 3' to 5'. It's the big gray structure in this figure. And on the side that it can go 3' to 5' and fit in there, it just makes one continuous strand. On the other side, because it can't fit in there, it does little snippets, keeps backing up, and then those snippets are connected with the ligase.